I've been interested in genetic phenomena since I was in high school. I'm mostly involved in genetic research using rice. Normally, efforts at crop improvement are aimed at increasing food production. But in this Moonshot project, the theme is development of biomass crops that can contribute to resource circulation. We begin by focusing on specific genes involved in biomass productivity, specifically photosynthesis. Using a new technology called genome editing technology, we edit genes into the ideal form for increasing biomass in the target cultivar. Today, we're hearing calls to develop technologies for capturing CO2 from the atmosphere. Normally, crops use photosynthesis to capture CO2 from the air, and the carbon in the CO2 is fixed as biomass. This natural process indicates the strong possibility that biomass can be used for resource circulation. Whereas conventional plant breeding aims to increase crop yields, our project must have one additional purpose: to produce materials for resource circulation. Our objective is a new type of improvement, one that achieves a quantum leap in the ability of crops to produce biomass. We aim to achieve both increase in food production and production of materials for resource circulation. For our research project, we chose 3 crops: rice, maize, and sorghum. In the case of rice, for example, we're focusing on 3 factors for biomass production: photosynthetic capability, ability to capture nutrients, and number and size of rice grains. Targeting the genes involved in these factors, we're using the genome editing technology to customize the genes in an ideal configuration for our purposes. We do this to create rice with high biomass productivity. For maize and sorghum, instead of genome editing, we use a phenomenon called heterosis, also known as hybrid vigor or outbreeding enhancement, to try to improve the crop variety. We are attempting to increase biomass to extreme levels while actively using wild strains, which are difficult to handle using conventional heterosis. Our current goal is to double biomass productivity from its current levels. In practice, however, increasing yield by changing only a single gene is no simple feat. In this project, we thought it would be technically interesting to increase biomass all at once by altering multiple genes at the same time. Rice farming has flourished in Japan since ancient times. Japan has a long history of active development of paddies for rice production. Today, however, only about 60% of Japan's rice paddies by area are in use. We aim to develop new and improved crops needed to restore the environment and introduce them into unused agricultural land, fallow rice paddies. Our expectation is that this effort will lead to stable food supplies and contribute solutions to any future problems of food security in Japan. By developing crops that dramatically increase biomass production capability while capturing and fixing CO2 from the atmosphere, we aim to achieve direct air capture agriculture that contributes to carbon resource circulation.
NEDO